Welcome to the Amgen Biotech Experience program. My name is Alia Katarna, and I'm the site coordinator for ABE Massachusetts. This program is a wonderful opportunity to improve teachers and students' knowledge about biotechnology. We understand the time and effort that participation in ABE requires and appreciate the commitment that teachers make to enhance their students' educational experiences. The ABE laboratories allow students and teachers to become immersed in key concepts and techniques in biotechnology. This series of laboratories has been carefully designed to reflect the research and development process at companies like Amgen. During these laboratories, students will gain firsthand knowledge and experience in the techniques used to produce therapeutic proteins such as insulin and human growth hormone. Participating in ABE will allow students to see their experience in science class as more real and relevant. ABE Labs involve students in teamwork and engages them in problem solving and critical thinking. These laboratories provide a unique opportunity to model the cooperative and challenging nature of the biotechnology industry. ABE teachers can choose which sequence of laboratories best suits their curriculum needs. They can choose between the complete sequence or the abridged version of the laboratories. During Laboratory 1, students learn how to use basic biotechnology tools. In Laboratory 1.1, students are introduced to micropipettes, which are used to measure and transfer small amounts of liquid. In Laboratory 1.2, students are introduced to gel electrophoresis, which allows for the separation and identification of small biomolecules. These tools are key in biotechnology research and development. Proper use of these tools is crucial for student success in the remaining laboratories. During Laboratory 2, students will learn how to manipulate plasmids using restriction enzymes to isolate the gene of interest. Using these two key tools, students will prepare for engineering the plasmid with the gene of interest in Laboratory 3. Laboratory 2A of the abridged sequence allows students to digest one plasmid to reveal the gene of interest, but they do not carry out the ligation using a second plasmid and additional enzymes. DNA ligases can join the ends of the restriction digest fragments to create plasmids with the desired RFP and antibiotic resistance genes. Laboratory 3 allows students to join the fragments they produced in Laboratory 2 and create a plasmid that has all of the correct components needed to produce the protein of interest in Laboratory 5. During Laboratory 4, students employ the technique of gel electrophoresis in order to verify that they created the correct plasmid during Laboratories 2 and 3. If students are doing the abridged sequence, they will confirm that they were given the correct plasmid before transforming bacteria to produce the protein of interest. During Laboratory 5, students will use changes in temperature and pressure to get their bacterial cells to take up the desired plasmid, therefore transforming the cells. Those cells with the correct plasmids will carry the antibiotic resistance gene along with the red fluorescent protein, allowing for selection of transformed bacteria by using antibiotics in the growth media. During Laboratory 6, students will isolate the desired red fluorescent protein from the transformed cells. Using column chromatography, they will purify the protein of interest, mimicking the last step in the production of therapeutic proteins. Students will utilize another essential technique of biotechnology, polymerase chain reaction, to make copies of plasmid fragments and further verify that their laboratories were successful in properly creating plasmids and transforming their bacterial cells.